Hi, everyone. And yeah, thank you for joining us today to hear a little bit about our work on what it says there, the composition, organization, and function of living membranes. So to kick it off, I'd like to tell you a little bit about why we're so excited about membranes in the first place. And that is that I happen to think membranes are fantastically interesting from the point of view of biochemistry, biophysics, cell biology, and material science. So it should be self-evident, but just in case it isn't, membranes are self-assembled materials, right? So lipids will spontaneously come together to form these materials that are unlike almost anything else in nature. At the two-dimensional or at the molecular level, they are quasi-two-dimensional fluids with practically no stable interactions between any of the components. But viewed at larger length and time scales, they behave very much not like fluids, but rather like robust elastic sheets, as you're seeing here in this movie of an isolated plasma membrane being manipulated by a micropipette on one side and an optical tweezer trap bead on the other. So clearly, a cup of water would not behave like this. This behaves very much like a solid, relatively elastic balloon. Zooming back in molecular scale, the physical and chemical gradients that are inherent to lipid membranes are, again, nearly unmatched elsewhere in nature. If you imagine, say, a chemical probe passing through the interface of the bilayer, you would go from an environment that's exclusively aqueous to one that's almost exclusively apolar in a length scale of a few angstroms, really. So you can just imagine the gradients that such a probe, or rather a transmembrane protein, would have to experience. And the same is true of physical forces. Hundreds of bars of tension at the head group level are precisely balanced by hundreds of bars of pressure as the lipid acyl chains are entropically pushing on each other to try to get away. But membranes aren't just fascinating and beautiful. They are also extremely important biologically. And I think that's most easily underlined by pointing out the fact that something like 1/3 of the mammalian proteome encodes for transmembrane proteins. That is, proteins that are forever, throughout their lifetime, embedded in a lipid bilayer of one sort or another. And that 30% doesn't account for the many other proteins that stably associate with membranes through lipid binding domains, through post-translational lipid modifications, through alpha helical amphipathic domains, and through direct interactions with other membrane proteins. So I think it's fair to estimate that being conservative, a major fraction, being a little bit flamboyant, I'd say that the majority of all bioactivity in the entire biosphere occurs either within or very close to a membrane interface. But these membranes aren't just a host for all this activity. They're also, in many important ways, a guide. And what I mean by that is, in almost every context where it's been carefully studied, the structure and function of membrane-embedded proteins is dependent on the composition and structural properties of the bilayer by which they're solvated. So from membrane receptors to transporters, ion channels, membrane-bound enzymes, and really most generally, the organization and structure of transmembrane proteins are starkly and sensitively dependent on both individual lipids in membranes and the properties of their collectives, things like membrane thickness, membrane fluidity, and others. At the same time, we've come to realize that mammalian membranes in particular are much more complex, more diverse, and more plastic than was previously appreciated. And really, these insights have been enabled by methodological developments in lipidomics, like the ones I'm going to talk about, in part led and mediated by lipotype, that have revealed that any given mammalian membrane may contain hundreds, if not 1,000 or more, individual lipid species, and many of these are regulated during various cellular processes, and that's really going to be the stories that I'm going to try to tell you today. So that's complexity. What I mean by diversity is that when we've begun to measure lipid compositions across various physiological contexts, we find that they vary rather dramatically. For example, here in cells isolated from representative of different tissues, here leukocytes, neurons, and epithelial cells vary dramatically in the composition of both the hydrophilic head groups of lipids as well as the length and unsaturation of the hydrophobic portion of these lipids. The details here aren't important. I really just show this data to emphasize the kind of diversity that exists between cells within an organism. And here, even what I'm calling the neuronal lipidome isn't something that persists throughout the lifetime of an organism. So as an example here, these are showing lipidomic trends through the first few weeks of life. So these are isolated neuronal plasma membranes from mice shortly after birth versus a couple of months after birth. And again, what I hope that you can see are sort of progressive changes, both again in the head group composition and in the hydrophobic acyl chain composition in these animals as they're developing. And finally, about plasticity, we've come to learn that membranes are remarkably susceptible to a variety of exogenous inputs, most particularly from dietary fats. So things like saturated lipids, polyunsaturated lipids, and cholesterol can be incorporated in toto, right, without being metabolically disassembled into the membranes, into the lipids that comprise our membranes. And that leads to dramatic compositional changes, which I'll tell you about in the first and somewhat in the second part of my talk. So if you believe me, that membranes are extremely diverse, plastic and complex, while also being super important for cellular physiology, then this raises, I think, a grand challenge, both for our lab and more broadly for the field, to understand the mechanistic connections between membrane composition, the biophysical properties that result from these complex collectives of lipids, and ultimately how composition and membrane structure combine to yield the functional membrane phenotypes in living cells. So this is what our lab tries to do. This is what our lab is focused on. And within this broad context of the nexus of membrane biology, I intend to tell you today, hopefully I'll have time, to touch on 3 different stories from the lab, each of which I think touch on different aspects of that nexus. Okay, so rolling right into story number 1, I'd like to tell you a little bit about our studies on the differentiation of the plasma membrane phenotype. So in addition to all those other amazing things that I told you about membranes earlier, mammalian plasma membranes have this amazing capacity for spontaneous lateral self-organization. So not only do they self-assemble, but they can self-segregate into coexisting liquid domains. And that's what you're seeing in these videos. These are plasma membranes isolated from a cultured mammalian cell and then stained with a fluorescent lipid. And what you can see is that over time in these movies, dark domains develop that exclude this fluorescent lipid, and we can tell that also that these domains are both fluid because of how they move and shape and change their shape. So in the physical chemical principle behind this behavior is simply that some lipids like one another more than they like other lipids, which is to say saturated lipids and sterols and glycolipids and sphingolipids all prefer to play together and exclude the more floppy-tailed unsaturated lipids. The differences in these binary interactions, a preferred saturated to cholesterol interaction versus a dispreferred saturated to unsaturated interaction, are actually quite small. All lipids like to stick together into membranes, but the inherent collectivity of membranes multiplies those small differences to produce large-scale organizations, like in those movies that I showed you on the previous slide, or here in simulated membranes, in synthetic membranes, and indeed in living membranes. We can observe large-scale, macroscopic, sort of micron-scale separation of coexisting ordered and disordered domains. And in cells, this capacity for spontaneous self-organization into coexisting ordered and disordered domains is functionalized and goes by the somewhat cartoony name of lipid rafts. So the idea here is that the self-assembly of certain lipids and proteins into a relatively ordered membrane domain can organize proteins laterally and regulate their interactions, their conformations, and ultimately their cellular functions. And the way that we really know that that this is important, that this behavior is important in living systems, or at least one way that we know that, is through studies of one of my favorite model systems, which I'm going to introduce to you briefly here, and they're called giant plasma membrane vesicles. GPMVs are basically plasma membrane blebs that can be induced by sort of mild chemical torture of cultured mammalian cells. That's what you're seeing here, the last hour in the life of these poor cells, which have been poisoned with maleimide, a chemical called maleimide. When we do that, for some reason that remains mysterious, the plasma membranes of these cells inflate, and eventually these bubbles of plasma membrane, these balloons of the plasma membrane, will pop off the surface of the cells, where they make a beautiful biochemical preparation that's really perfectly clean of almost all the common contaminants in membrane preparations. So these are really isolated mammalian plasma membranes from a particular cultured cell type. Equally interesting, that's from the biochemical point of view. From the biophysical point of view, equally interesting is that these isolated plasma membranes undergo spontaneous phase separation into coexisting ordered and disordered phases in accordance with the lipid raft hypothesis. And what I mean by that is that sterols and saturated lipids and GPI anchored proteins will co-segregate away from a relatively disordered phase that is comprised largely of unsaturated lipids and other proteins. So this model system allows us to investigate the biophysical and biochemical properties of plasma membranes from mammalian cells in this kind of isolated, well-characterizable system. And the first question that we got into using this system is introduced here. So from a variety of classical studies reviewed in this classic review, we know that the plasma membrane is distinct from and really unique to other membranes in a mammalian cell. It's highly enriched in sterols and saturated lipids, in charged lipids, etc. But the question that we started to ask is, are plasma membranes from different cells different, right? So until now, I've talked about membranes, or at least plasma membranes, as this one monolithic thing, But do we have any reason to think that a plasma membrane from one tissue or one cell type would be identical to another? So for example, if we compare two different types of blood cells, or a stem cell to a differentiated cell, or two sort of lineage related cells, would we expect their plasma membranes to be identical? And in particular, these last two comparisons are relevant for the specific model system that we've chosen to test this question, which are mesenchymal stem cells. So mesenchymal stem cells are primary cells. We actually use human cells from bone marrow donors. You need very little, and they're easy to expand. So they're a beautiful stem cell type to work with. And also in culture, they can be induced to differentiate into one of several morphologically and functionally distinct differentiated lineages. In particular, in this study, we focused on osteoblasts, which are bone depositing cells, and adipocytes, which are lipid storage, fat storage cells. And that's what you're seeing in these images. And we can literally see, for example, here by staining for calcium, we can start to see calcium phosphate being deposited by, basically pre-bone being deposited by these osteoblasts, or these adipocytes swelling up with massive amounts of lipids, or these sort of boring fibroblast-looking stem cells. So the obvious advantage of a culture system like this is that with nearly identical culture conditions and entirely identical genotypes, within a week or two, we can generate three completely different, again, phenotypically different cell types, and then ask, are their plasma membranes identical to one another? And the answer, as you might not be surprised to, as I'm telling you the story, is they're dramatically different from one another. So I'm not going to belabor how exactly we characterize these various parameters. You can just believe me that we can use these giant plasma membrane vesicles, these isolated plasma membranes, to evaluate, for example, how stable lipid rafts domains are in various plasma membranes. And what we see is that differentiating mesenchymal stem cells into osteoblasts or adipocytes dramatically changes this biophysical parameter, this raft stability. And not only are the differentiated cells different from the stem cells, but they're dramatically different from one another. The direction changes. Here's another example, an unrelated, a totally distinct but possibly related physical property, which is the overall lipid packing in these plasma membranes. Again, changes dramatically during cellular differentiation, with osteoblasts showing tighter lipid packing and more stable membrane microdomains relative to either the undifferentiated cells or especially the adipocytes. So that led us to the obvious question. As membrane biophysicists, it was the most obvious prediction that if these plasma membranes have different biophysical properties, they must have interestingly different compositions. And indeed they do. So we partnered with Lipidomics. This was many years ago now, even when I was still back in Dresden, to measure the lipidomic composition of the plasma membranes and the total membrane samples isolated from MSCs or from these differentiated cell types. And what you're seeing in this heat map, blue represents individual lipid species that were reduced during differentiation into adipocytes, and vice versa for red and osteoblasts. So these lipidomic barcodes are pretty to look at, but hard to get anything useful out of. So ultimately, we have to kind of recombine this data into something sensible that we can understand from a biological point of view. Specifically, we can use statistical approaches like principal component analysis and find that, for example, in this case, osteoblasts seem to have 3 different human donors of osteoblasts. And again, these are not osteoblasts, these are mesenchymal stem cells differentiated in vitro. into osteoblasts showed a completely distinct and specific lipid signature from either the adipocytes or the undifferentiated mesenchymal stem cells from which they were born. So not only can we see that there's this consistent differentiation of the plasma membrane lipidome, but also we can define which lipid species define the osteoblast lipid signature. And specifically what you might see here is this notation, which denotes the total number of carbons in the lipid acyl chains and the total number of double bonds within those acyl chains. And what you'll see here are relatively big numbers, and here you'll see relatively small numbers, which means that the osteoblast lipid signature is defined by relatively long and relatively polyunsaturated glycerophospholipids. And again, we can now break this down into individual lipid components and really look more directly and see that, yeah, the PCA didn't lie to us. The loadings plot showed us exactly what was different. And it showed us that osteoblast plasma membranes are characterized by relatively long and relatively highly unsaturated lipid acyl chains. So that was neat and a surprise, and we had no idea what to expect, but we were glad to see something interesting. In particular, this started us down the path of looking at the longest and most polyunsaturated lipid components present in mammalian cells, which are omega-3 polyunsaturated fatty acids like DHA and EPA. These two are the main components of fish oil. These are common supplements. These are things that are meant to be good for our health for reasons that are confusing, although they are remarkably true. Since then, there's been some really interesting epidemiology done that shows that high levels of omega-3 polyunsaturated fatty acids, not just in the diet, but actually in patients, are massively protective against overall mortality. Something like 15% less overall mortality in the highest quintile, I believe. Anyway, email me if you want the reference. But exactly according to expectations, with osteoblasts being relatively high in long and polyunsaturated lipids, they were also high in these long polyunsaturated lipid components, that is, these omega-3 DHA and omega-3 EPA. So wrapping up that very first bit, I hope that I've convinced you that osteoblast plasma membranes have distinct biophysical properties and unique compositions, being relatively enriched in these specific interesting lipid components. So that leads to many questions, many of which we asked ourselves and try to answer. Say why, right? Why would these cells during in vitro differentiation have developed such a unique plasma membrane phenotype? The answer to that is I don't know. But the function of these osteoblasts may have something to do with the unique demands on their membranes. Specifically, osteoblasts secrete bone in a sense. So they secrete large collagen proteins. They probably secrete other factors that are important for making bone. And perhaps this puts unique demands and stresses on their secretory system, which are reflected in their unique lipidomes. Okay, so that's something that we one day want to study, but not right now. So now, instead, we asked whether MSC differentiation is sensitive to the plasma membrane phenotype. The thinking behind that was if during, say, osteoblastic differentiation, these cells are acquiring a particular plasma membrane phenotype, then it suggests they need it for something. And maybe there's some sort of checkpoint where these cells know that they have prepared and developed an appropriate plasma membrane phenotype that will allow them to be osteoblasts. And if we can inhibit or promote that plasma membrane phenotype, maybe we can interfere with, or again, promote differentiation down a specific lineage. And to do that, I'd like to point out that in the case of lipids, and only in the case of lipids, it's actually true that you are what you eat. So just to belabor that for a moment, whenever this, whatever it is, eats this hamburger, the DNA of the cow that is part of the burger will not become any part of the DNA of this critter. And the same is true for the proteins in the wheat or the cow. They will be broken down into their individual components and recombined into whatever proteins this creature needs for its life. But in the case of lipids, it actually is the case that the lipid components of our diets are indeed incorporated into our membranes. I showed you this cartoon earlier, but you don't have to believe my cartoons. I have data to prove it. For example, first ex vivo, so in cultured cells, we can feed this fish oil component, again, this DHA that I introduced to you earlier, to cultured mammalian cells, and a large fraction of all these cellular membranes will start to contain this fatty acid where they didn't before. Perhaps even more amazingly, we can do the same thing in living animals. We can take mice and let them free feed on a diet rich in fish oils, which contain these omega-3 polyunsaturated fatty acids. And these studies were all done in collaboration with a really excellent lipid scientist and biochemist, Rob Chapkin at Texas A&M. And Rob's data showed us that these animals, which normally have almost none of these kind of fatty acids in any of their membranes, after free feeding on a diet enriched in omega-3, in fish oil, which is itself enriched in omega-3 DHA, some 30% or more of all the lipids in their cardiac tissue now contain a fatty acid component like this, that these animals themselves, first of all, cannot make, because they don't have the enzymes for making omega-3 polyunsaturated fatty acids, nor do they very often see it in their diet. So this entirely exogenous lipid source is happily used by these animals to make up a very large fraction of the entire architecture of, here in particular, their cardiac tissue. So that's, I think, pretty amazing and surprising, but also a very nice tool that allows us to manipulate lipid composition either in culture or in vivo. So that's exactly what we did. I'll remind you, we found that osteoblasts, in particular their plasma membranes, are relatively rich in lipids that contain long polyunsaturated fatty acids like DHA. So the prediction was that feeding mesenchymal stem cells with DHA would reproduce a osteoblast-like plasma membrane composition, and maybe also the resulting osteoblast-like biophysical properties. And again, that's exactly what we saw. So here on this PCA plot, it's not super easy to interpret, but what these arrows show is how the overall lipidome changed with DHA feeding. And as you can see, in each direction, they changed in the same way, because this PC2 probably contains long polyunsaturated fatty acids, long polyunsaturated lipids. But more importantly, this movement along PC2 and a little bit down PC1 landed the undifferentiated mesenchymal stem cells right smack dab next to the osteoblast. That is to say, we could reproduce the osteoblast plasma membrane lipidome just by feeding DHA to MSCs. And consistent with that, we also observed some of the same biophysical changes that we observed during osteoblast differentiation. So here, for example, looking at microdomain stability from the undifferentiated cells changing into osteoblasts, DHA alone could stabilize membrane microdomains exactly like osteoblastic differentiation did. And this is just a cool molecular dynamics movie to show the effect of a membrane enriched in omega-3 polyunsaturated fatty acids and how we can directly visualize the stabilization of phase separation in membranes that are rich in omega-3 DHA. Okay, so summarizing, supplementation with a lipid component that characterizes osteoblast plasma membranes could induce an osteoblast-like plasma membrane phenotype, both compositionally and biophysically. And now the question is, would that actually affect anything to do with these differentiation of MSCs? And, you know, again, I probably wouldn't have pitched it like that if it didn't. So here you see the same staining that I showed you before. This is alizarin red, staining for insoluble calcium deposits, hydroxyapatite and calcium phosphate. And, you know, this image sort of speaks for itself. And we can quantify this osteogenic deposition, actually not just by image processing, but actually by extracting these stains, and show that quantitatively DHA supplementation in culture dramatically enhances osteogenic differentiation in these mesenchymal stem cells, you know, with the control, you know, compared to, say, a mono unsaturated fatty acid feeding at the same concentration. Okay, so, you know, that's sort of the pitchy conclusion, that we can re-engineer the plasma membrane phenotype by dietary lipids in order to, you know, get the particular effect that we might have been looking for, in this case, to potentiate osteogenic differentiation. So now a really big open question is, how does this work? What's the mechanism? And I'm going to tell you right now that we don't really know the answer. The answer is it's extremely complicated, right? I told you that a third of all proteins live in membranes, and many more associate with membranes. And it's possible that any of those, and maybe all of those, are affected by changes to the plasma membrane composition and plasma membrane physical properties. So at first we took a kind of a basically unbiased approach and just did a proteomic screen of various important signaling proteins and their post-translational modifications. And actually this turned into a kind of a nice and surprising effect, where the top two hits of signaling modifications that went up with DHA feeding and osteoblast feeding were phosphorylation and activation of AKT, which is a plasma membrane resident kinase, also known as protein kinase B, that's important for osteogenic differentiation. So I'm not going to go too deep down this path. If you're interested, you can check out this publication. And what you'll find is that we claim, to summarize this part one, is that re-engineering of the plasma membrane phenotype by dietary lipids can affect cell function. In particular, in this context, that we could potentiate osteogenesis in mesenchymal stem cells by remodeling plasma membranes with DHA. And the mechanism is, it's complicated, and I don't want to get into it. Okay, so the second story kind of evolved directly from this first story, which really came from looking more deeply at the lipidomic effects of DHA feeding. What we saw is, this is the slide I showed you earlier, where omega-3 polyunsaturated fatty acids are dramatically incorporated into membrane lipids. We could also see the same thing with this omega-6 PUFA, arachidonic acid. But if we look at the whole lipidome, we saw a lot of other interesting stuff. And in particular, that's summarized here. So what you're seeing here is a graph of the changes to total lipid saturation, right? The number of double bonds between the two acyl chains after feeding cells with either arachidonate in blue or DHA in purple. So I'll just walk you through some of the stuff that we saw and get to the most surprising and interesting one. So the first thing you see is that when you feed the 6 unsaturated DHA, we get a lot of 6 unsaturated lipids. That's basically here. You also have lipids with even more unsaturations, either because this was modified by adding another one, or more likely because the second acyl chain was also, say, monounsaturated. Then the same thing with the arachidonic acid. you have 4 double bonds and 5 double bonds increasing. Okay, that's exactly what we expected. That's the incorporation of these fatty acids into lipids. Also pretty reasonable and could easily be explained is that other polyunsaturated fatty acids went down, right? So the way that we would explain this is that DHA, say, is being incorporated in place of a 3 or a 2 unsaturated acyl chain. And so these things are going down while these things are going up. But really unexpected, and I think quite surprising still, is the observation that fully saturated lipids were doubled or even increased by more than that in the case of both feeding these 2 polyunsaturated fatty acids and changing their composition. And this is for CHO cells, kind of a model cell line, but we observed this across all of the samples we tested, including all those MSCs that I showed you earlier, other random cultured cells, and also primary neurons. In all of those cases, we observed DHA incorporation, but also concomitant reduction of other polyunsaturated lipids, and again, this dramatic increase in fully saturated lipids. And finally, going back to our samples from our collaborator Rob Chapkin, we could see this again almost doubling of fully unsaturated lipids in these animals that were fed with and incorporated to a dramatic extent 6 unsaturated DHA, probably from these fish oils. And finally, just because we could, and because we wanted to know, we measured the temporal profiles of these, both the incorporation of these exogenous fatty acids and the lipidomic compensation that I showed you earlier. So here you see that after adding DHA to these cells, it was incorporated relatively quickly. The half time is approximately 4 hours for the sort of full lipidomic turnover. And nearly concomitant were these other changes. The other polyunsaturated lipids going down and the saturated lipids nearly doubling were practically happening almost at the same time with a very, very small lag. So that's what it says there. Saturated lipids increase concurrently with DHA incorporation. So what's going on here? To explain it, I'd go back to some of the really basics of membrane biophysics. One of the first stories that got people excited about membrane biophysics was this sort of thinking that membranes in cells have a variety of demands placed upon them. They need to be robust, impermeable, relatively rigid, but at the same time, they have to be fluid and flexible to allow all of the biochemistry that needs to happen. And this is enabled by membranes, lipid bilayers settling on a relatively narrow sort of sweet spot that balances these competing demands of keeping fluidity high, but not too high, and permeability low, but not too low. And importantly, membrane properties are highly influenced by both endogenous and exogenous inputs, like, for example, temperature most easily, right? So if we lower the temperature of a particular lipid bilayer, say you're an E. coli and it gets dark and cold, then your membrane lipids will stop flopping around, they'll slow down, and they'll start to get more rigid and more tightly packed. And after a while, these membranes will fall out of their functional zone, and in this case, become possibly too rigid for life. The reason that they don't become too rigid for life is that organisms from E. coli to yeast, and even in certain mammalian species, it's been shown that there are compensation mechanisms to restore functional membrane properties to compensate for changes in the environment. So for example, if we go back to our E. coli example, they get cold, their membranes get too tightly packed and too stiff, And these cells can sense that effect and upregulate enzymes that will produce relatively unsaturated acyl chains, relatively unsaturated lipids, and restore baseline membrane fluidity now at a new lipid composition. And we claim that something like this is also happening in our mammalian cells, that when we change their not now temperature, but diet by addition of these highly polyunsaturated fluidizing fatty acids, they compensate by removing other unsaturated fatty acids and instead upregulating highly saturated lipids to recover their membrane fluidity. And we can test this hypothesis by biophysical experiments using reporters that can directly or somewhat directly report on membrane physical properties. In particular, the one that we relied on for these experiments is called Lordan. It's a fluorophore that changes its emission spectral characteristics depending on the properties of membranes in which it's dissolved. So in relatively tight packed membranes, it tends to be more blue. In loosely packed membranes, it tends to be more green. And this in color here is essentially the ratio between the blue and the green signals, with warmer colors representative of more tightly packed membranes. And you kind of have to squeeze to see the effect, but within these dotted lines that we sort of tried to capture most of the internal membranes in these cells, I hope you can see it's sort of going from this teal color to this dark blue to this dark blue to this teal again. And what we see and what we think we're seeing is that shortly after addition of this polyunsaturated fatty acids, the membrane packing is rather significantly decreased in these cells. But then after a time, which actually coincides well with the time of lipidomic compensation, we see that the biophysical properties, that is lipid packing in these membranes, is restored. So that's the idea, right? These lipidomic compensation is effectively a biophysical compensation to restore membrane fluidity back to baseline after this perturbation with polyunsaturated fatty acids. So, but what challenged that hypothesis or that interpretation is that we could look at the total unsaturation in the membranes and see if indeed it was restored, right? So we got a bunch of polyunsaturated fatty acids. We have fully saturated lipids on top of them. Maybe the unsaturation was restored, but it wasn't. In that these fed cells, which biophysically looked like they went back to normal, compositionally were clearly not normal, were clearly much more unsaturated. So what else is happening? Well, the answer is actually pretty simple. Mammalian cells have another knob that they can turn to tune their biophysical properties, and that knob is cholesterol. So cholesterol is known to lead to tight lipid packing, and consistently, we saw cholesterol highly upregulated in these DHA-fed cells. And again, we could look at the time course of this upregulation, and again, it was nearly concomitant with the time course of DHA incorporation and those other lipidomic changes. And finally, going back to the tissues from the animals, we could see that cholesterol was upregulated in those as well, right? So this is going to be membrane cholesterol as a percentage of membrane lipids being highly increased by these fish oil-rich diets. So that's cool, both because cholesterol is super interesting and very important pathophysiologically, but also because a variety of methodological tools have been developed to probe cholesterol synthesis and trafficking, and these allow us knobs to turn in order to manipulate this lipidomic compensation response that we see. For example, there's an enzyme in the Golgi called Site1 protease, and that is essential for releasing a transcription factor called SREBP, which goes to the nucleus and makes target genes, which ultimately make cholesterol and cholesterol trafficking machinery and lipid synthesis machinery. This SRABP is really an essential factor for these, for lipogenic gene expression, and also for the compensation that I showed you earlier. So when we have cells that are deleted for this S1P, SRABP cannot be activated. And also, consistently, we see that this DHA-mediated increase in cholesterol synthesis is abrogated. We can also inhibit SRABP in a different way, pharmacologically instead of genetically, by this compound called betalin, which somehow blocks the trafficking of these cholesterol sensing machineries to the Golgi, where they meet their site 1 protease for release. And again, pharmacological inhibition of SRABP, again, abrogated this DHA-induced increase in cholesterol. Perhaps unsurprisingly, but validatingly, the biophysical compensation that we observed in normal cells, where DHA first led to a reduction in lipid packing and then a recovery, under pharmacological inhibition was inhibited. And again, under genetic inhibition of this response was again inhibited. So these cells now have more fluid membranes that cannot recover. Now, I showed you earlier that life depends on this very subtle balance. And the presumption is that these cells would now have that balance thrown off by the addition of an exogenous polyunsaturated fatty acid, and then the inability, if we use one of these inhibitors, to recover their normal plasma membrane phenotypes. So now the question is, does screwing around with their membranes affect something about cellular fitness or behavior? And again, obviously the answer is yes. So these are curves showing the effect on cell viability of DHA feeding in normal wild type CHO cells, or these cells that have a deletion for the SRABP processing enzyme S1P. And what I hope you can see is that even very low levels of DHA will kill these cells under conditions where wild type CHO cells are perfectly happy. And again, with pharmacological inhibition of this response, we again see a dramatic reduction in viability in these cells. So summarizing part 2, what we call homeostatic membrane adaptation, we see perturbations of membrane lipidomes by dietary fats that induce a robust compensatory response to maintain membrane packing. And inhibiting this response is toxic. And so what we conclude from this is that homeothermic animals like us and other mammals have co-opted the thermodaptive machinery of cold-blooded animals or simple organisms like E. coli and yeast to cope with dietary inputs, the way that these animals cope with temperature perturbations to maintain membrane homeostasis. And I'll wrap up by thanking the brilliant group of people that it's my pleasure and honor to work with every day. In particular, I have to highlight Dr. Candice Levental, who is the co-PI of our lab. And she did a lot of the work that I showed in the first 2 parts of the talk. Joseph Laurent, who did all of the asymmetry work that I didn't get a chance to show. And actually, many of these people have contributed throughout. Some of our favorite collaborators, one of which I mentioned. And thank you for your time and attention. I hope I've left some time for questions.